Welcome, everyone, to this week's edition of the Extracellular Vesicle Club. I am very happy to introduce Professor Jun Yamashita of the University of Tokyo and some colleagues who are going to be joining to present an exciting paper that has been pre-printed in bioRxiv recently. I'd also like to thank Stefano Pluchino, who made the suggestion of this paper. So I know Stefano is someone who is very widely read, has a lot of insights, and I'm always appreciative of his suggestions because we, as scientists, I think we all always like to think that we're very smart and we know everything that there is to know about the world around us, except for maybe a few little things that we're trying to put together. But sometimes a paper comes along that reminds us of how little we might actually know and how exciting it really is to be working on the world of biology. And I believe that this paper that we're going to be discussing today could very well be one of those discoveries. And we're going to be talking about some vesicles. This is, after all, the Extracellular Vesicle Club. And whether they're extracellular vesicles or not, that might be something that we can decide in the course of this journal club today. But in any case, definitely some new and interesting findings here. So before we get started, I'd just like to remind everybody to please put your questions and your comments in the chat box. And although you will be on mute during the presentation, at the end of the presentation, I'm going to go through that chat box. I'm going to allow unmuting, and you can then have this conversation with Professor Yamashita and his group. So I think that's all I need to say. So let's get on to the exciting science. And Jun, Professor Yamashita, please go ahead.
Yeah, thank you so much for that very kind introduction from Dr. Witwer. So, hello everyone. I am Jun Yamashita from the University of Tokyo in Japan. Thank you for joining us at this possibly inconvenient hour. I truly appreciate your time. I'm honored to be here and would like to thank Dr. Ken Witwer for generously providing me this precious opportunity to present our recent work. And I'm also very grateful to Dr. Pluchino. He found our reprint on bioRxiv and posted Dive into Dive on X and encouraged today's presentation. So thank you so much. And I look forward to sharing our data with all of you. Okay, well, can you see our slide and pointer?
Looks good. Everything looks good.
Okay. Okay, let's get started. Okay, today I like to talk about two main points. First, I'll give some background on how we reached the discovery of DIVE, and primarily that is through the discovery of psych phenomena, phenotypic synchronization of cells. But this background might be a little bit long, but I hope it helps your understanding on our work. Second, I'll discuss our finding regarding DIVE itself. Okay, during the embryonic development, cells must differentiate in a coordinated manner. Sufficient amount of cell mass with the same phenotype appearing synchronously would require to normal embryonic development. If cell-cell communication for the synchronization is not existed, each cell would differentiate into different cell types that would result in improper development. But so far, how cells synchronize their differentiation stages with each other remains largely unknown. We got a chance to approach such question through our research on stem cell differentiation. Let me briefly share our earlier work. We have been investigating about pluripotent stem cell differentiation mainly to the cardiovascular cells. We first induced frequent positive mesenchymal cells from ES or iPS cells, and we further induced various kinds of cells, endothelial cells that form the vascular tube, and mural cells that is like smooth muscle cell-like cells that support the blood vessel structures, and blood cells flow inside of the lumen, and cardiomyocytes pumping out blood. And all these cardiovascular cells can be systematically induced from the common progenitor frequent positive cells. Okay, we generated three-dimensional vascular structures in vitro using this system. We formed aggregates of the African positive vascular progenitor cells and cultured them three-dimensionally in type 1 collagen gel. After 3 to 5 days, cells sprout out and generate tube-like structures. Immunostaining confirmed that these structures consisted of the endothelial tube, colored purple, and mural cells attached from the outside, colored brown. And also we confirmed blood cells existed inside of the lumen. And this is a time-lapse video of the vascular formation in vitro. So you can see the free composite cell aggregate here. This aggregate grows keeping spherical shape in first days, but after that, many cells start to sprouting out from the aggregate. And these cells form the tube-like structure. Yeah, you can see here. And you can see, for example, sprouting of the vessels. And here you can see the endothelial cells moving from this tube to left tube and generate anastomosis, and finally vascular networks are formed like that. So we can generate the three-dimensional vascular structure in vitro. After this work has been done almost a quarter century ago, and consider I think that this is one of the earliest examples of organoid research, and at least this is the first vascular organoid generated from stem cells. But this is more important things, that is live imaging is critical in our research from long time ago. Okay, in our previous stem cell research activities, we found that activation of the PKA protein kinase A can accelerate the differentiation speed of the mouse ES cells. We generated a cell ESL9 PKESC, in which PKA can be inducibly activated. In this case, a DOX-off system, inducibly activated by a TET-inducible expression of the constitutive active form of PKA. And this is a time course of the PKE cell differentiation. Under the DOX plus control condition, frequent positive mesenchymal cells appeared around day 4 of the differentiation. Whereas under the DOX depleted conditions, PK was activated. In such conditions, frequent positive cells start to appear from day 1.5 and increase, and it becomes around 40% on the day 3.5. So using this cell line, we can intentionally manipulate the speed of differentiation. Okay, then we came up with a new question and corresponding experiments. So that is, if we co-culture this fast differentiating PKES cells with wild type ES cells, in this case, they just expressed GFP. Then activate the PKA only in the PKA ES cells. So these cells differentiate quickly. Then how do these wild type ES cells behave? So this slide is the answer. Under standard conditions, wild type GFP ES cells differentiate into frequent positive cells around day 4 or so. They are similar as the PK cells. And under the co-culture and depleted the DOX and activated accelerated differentiation, so PKAES cells differentiate into frequent positive cells around day 1.5 or so. And it's rather surprisingly, even in the GFP-ES cells, in the wild type cells, in which so PK was not activated. So differentiation speed was accelerated, and frequent positive cells start to appear around day 2.5, and by the day 3 or 4, they caught up with PK-ES cells. So this graph shows the percentage of frequent positive cells appearance over time when co-cultured with fast differentiating ES cells, wild type GFP cells, also speed up their differentiation and synchronize them with the PKE cells. So then we named this phenomenon phenotypic synchronization of cells, psych, in this case in differentiation. Okay, so next question is what type of signaling is involved in psych? Among various possibilities, we found that extracellular vesicles play a central role. First, we performed loss of function experiments for EV secretion. We treated cells with NSMS2 inhibitors, minimizing GW compounds, and we added these compounds to the co-culture system. And you can see how the accelerated differentiation of the PK cells was not disturbed, but only the appearance of the GFP positive cells, African positive cells from GFP positive cells are disturbed. So that suggests that EV is involved in the psych phenomenon. This slide shows immunostaining for FLIC1 and GFP, FLIC1 colored magenta. And you can see many white cells. These cells are double positive for FLIC1 and GFP. That means that these cells are synchronized differentiated cells from the wild type GFP cells. And when minimizing and GW compounds added this condition, these white cells are almost disappeared, and whereas the magenta color African positive cell appearance from the PK cells was still existed. This is indicating that the EV is crucial for the synchronization of the differentiation. To confirm the role of EVs, we collected EVs from the fast differentiating cells and added them to the differentiating wild type cells. And this is immunostaining for the FLIC1 mesenchymal cells and nano for the pluripotent stem cell marker. And as you can see here, when the EVs from fast differentiating cells added to the wild type cells, appearance of the FLIC1 positive cells accelerated. Thus, we demonstrate the psych phenomenon and the central role of EVs in psych. Okay, next we try to identify which molecules in EVs are responsible for the psych phenomena. We did not find any relevant proteins on mRNAs, so we focused on microRNAs enriched in EVs in the fast differentiating cells. And then we selected these three candidates MyRs. And we generated MyR overexpressing cells and co-cultured them with, in this wild type recipient ES cells. In this experiment, MyR expressing cells express GFP and recipient cell negative for GFP. And co-cultured them and examined the mRNA which induce mesenchymal cells in the recipient cells. We analyze only in the GFP negative cells. And then, as you can see here, among these 6 MyR candidates, MyR133 induced FLIC1 positive mesenchymal cells in recipient cells. Okay, this time I show just a summary of the results. So PK activation in the fast differentiating cells, inducing the early appearance of the FLIC1 positive cells, and also induce the expression of MyR133. And MyR133 is transferred to the neighboring recipient cells, is EVs. In the recipient cells, MyR133 suppress the reported target, SPRY1 and RAS1. In this case, RAS1 expression was reduced, and these things resulted in the reactivation of the pKa in recipient cells. That was confirmed by the expression of the phosphorylated crepe in the EV-added recipient cells. So thus, PK activation, the same situation in the donor cells that reproduced in the recipient cells. So we consider that this is at least one of the synchronizing mechanisms. Okay, let me briefly show how strong is the effect of the EVs. It can show how strong effect the EV can show. We performed the ex vivo culture of early mouse embryo E3.5 blastocyst and added EVs to them. And EV treatment can accelerate cardiomyocyte induction. Sometimes, whereas nearly no cardiomyocytes appear under control conditions, but with the addition of the EVs, almost all cells began to beating. So indicating that EV has a very potent effect that can even shift the cell fate towards cardiomyocytes. And I noticed that the psych effects are potent in when cells were co-cultured, but the effect is not observed when cells were separately cultured using the transfer membrane. So this result indicates that direct contact of cells is required for the psych phenomenon. Okay, in summary, we discovered the psych phenomenon and which requires direct cell contact. Okay, sorry, it was a rather long introduction, but in this way, we arrived at the starting point of our research on DIE. So psych appeared to occur between adjacent cells. So we thought we might directly observe the vesicle behavior. So to do that, we attempt live imaging to capture the vesicle dynamics. We first generated several cell lines that express fusion proteins of EV markers CD63 or CD81 and with fluorescent proteins such as EGFP or TD tomato. And in green cells, vesicles and plasma membranes appear green, while in red cells, they appear red. And by co-culturing these cell types, we try to observe the movement of vesicles between adjacent cells. This slide shows one of our early trials of live imaging. We co-cultured green and red cells, and here you can see several green vesicles observed existed in red cells and moving in the red cells. So actually, we can see that green vesicles in red cells, but due to low spatial temporal resolution, so we are not able to know the detailed dynamics at that time. And this is another preliminary images. So this is a boundary area of two cells, two left cells and right cells in the boundary of the cells. And we found that an array of the vesicles, green vesicles, lined up at the boundary region between cells. And that moved to the in this case rightward. So this suggests that vesicle may travel along some specific route between the cells. So, but under these initial imaging conditions, it was difficult to accurately track the details of vesicle movements. So we need some improvement of our imaging approach. The major breakthrough came from using bright and photostable fluorescent protein, stay gold, which replaces GFP for green label. So we use the stay gold instead of the GFP. And by combining stay gold with a high resolution microscopy such as ELIRA size, we could image the wider field with Z stacks in a short time. So in this case, such a three-dimensional image set can be obtained within 3 seconds. So these things allows us to track the fast-moving vesicles and capture their rapid dynamics. In this slide, we have two cells in center, two cells in center expressing CD81 stay gold, and surrounding cells are red cells, membrane stained in red. And as you can see here, numerous green vesicles observed in the adjacent red cells, indicating the predominant vesicle delivery to adjacent cells. This slide is a video recording the vesicle transfer between two mouse ES cells, cell A and cell B, that express CD63 stay gold, and membranes that are marked by the membrane staining called Kakshine reagent. And from the cell A, shown in the light blue, we selected the two vesicles, and you can see the two vesicles move to rightward and cross the cell boundary and moved apparently into cell B. So vesicles are actually transferred between adjacent cells. And this video shows that the co-cultured cells, green cells expressing CD81 stay gold, and surrounding red cells marked the plasma membrane with magenta color. And first, I'd like to show you the arrays of vesicles moving between two green cells in the center. So as you can see the many vesicles moving, and you can see the vesicles, array of vesicles move from left and right. And I suppose another sample you can see here, now vesicle array, now move from left to right. Okay, I'd like to show that again. Here you can see the moving of the vesicles, array of vesicles from left to right. And again, here you can see the array of vesicles moving from left to right. So now this video is compatible with the images I showed you just before, that array of vesicles seems to move between cells. Okay, next video recorded vesicle movement between red and green cells, red cells on the left and green cells on the right. And here is plasma membrane of red cells. Green cells express and extend vesicles. And you can see how many vesicles are moving both in red and green cells. And among them, we selected two vesicles, vesicle 1 and vesicle 2. Vesicle 1 moved towards the plasma membrane and attach and stay there shortly, and soon go into the red cell. Vesicle 1 move to the stay here shortly and soon go into the red cell. And another vesicles, vesicle 2, I'll show you that. Vesicle 2 has been already existed in the red cell, and first they move to the plasma membrane here, and soon it relocated around here and go and back sometimes, and finally go into the green cells. So green 2 first go to here like here, and relocate to the around here, and back and go, and finally go into the green cell. So yeah, you may notice that this place is something special. So yeah, I like to confirm that. So Vesicle one move to here and attached and go pass through the plasma membrane around here, yeah, around here. And Vesicle two come and go back, go and enter into the cells. So yeah, they use almost identical position of the membrane. So that suggests that there should be a special route, some specific route should exist around here. And these vesicles move very fast, at most around 1 micrometer per second, and pass through the very rapidly, pass through the plasma membrane very rapidly, just pass through the plasma membrane or, almost within 10 seconds or so, very rapid we pass through. And another important point is that even after the pass through the plasma membrane, these green vesicles are independently green, not accompanied by the red-colored plasma membrane components. So these things mean that such transfer is not mediated endocytosis. So from all these observations, we consider that this cell-cell communication will be the new modality. So we named this as a direct intercellular vesicle exchange. Okay, this slide we summarize the kinetics of these vesicles. So we named these vesicles intercellular vesicles, intercivils, and tracked trajectory. So trajectory revealed that the vesicle 1 and vesicle 2 move along almost identical sides of on the plasma membrane at speed about 1 micrometer per second. It's rather fast. And traversing membrane in this case within 10 seconds, and in this case within 20 seconds. So rapidly pass through the plasma membrane. Okay, next, we try to identify the structures that support the existence of type system with electron microscopy. Here we use the three-dimensional electron microscopy, PIPSEM. But even then, so as PIPSEM gives just a snapshot, so it is impossible to confirm, for example, whether a vesicle in the close proximity to the plasma membrane is actually crossing the membrane or not. It's impossible to judge that. So then, therefore, we focused on finding the arrays of vesicles in PIPSEM data. And this slide is one example. An array of 4 vesicles. It's around 50 nanometer diameter and with single membrane lined up across the plasma membrane. So three-dimensional reconstitution suggests that they may be traversing they may be passing through the plasma membrane. Okay, this is another example. In this example, so yeah, you can see now plasma membrane here, and in the next section, so plasma membrane signaling diminishing here, and there may be exist a pore-like structure maybe here, and the next section, so array of the vesicle-like signal observed like that. And in the next sections, plasma membrane go back to normal. So then how do we reconstruct this imaging in three-dimensionally? And as you can see here and here exists the plasma membrane and the pore-like structure, and vesicles seem to align across the pore, across membrane and pore. So from all these observations, now we hypothesize that type structures as follows. So plasma membranes of two adjacent cells fused and generate some pore and hole-like structure. And kind of filaments. I think that the actin fiber is most likely and extends through the pore. And vesicles are moving on the filaments driven by the molecular motor, such as myosin. And we consider this structure would be most resembled to length zero tunneling nanotube. So now I consider that this should be an putative dive machinery. Okay, finally, I'd like to shortly talk about the biological roles of dye. It's a recent bioarchive paper, but now published PNAS that reported direct contact dependent mRNA transfer between mouse and human pluripotent stem cells. So in this paper, the authors co-cultured mouse and human ES cells. It requires some explanation about that now. There are two pluripotent states, naive and primed. Naive is more immature state, and mouse ES cells are in naive state, and human ES cells are in primed state. And when these cells are co-cultured, so mouse ES cell-derived mRNA, such as TFAP2C or TFCP-KLF4, this mRNA is important for the naive status. So these mRNA are transferred to the mouse cells and induces a naive conversion of the human cells to naive state. So the author suggests that mRNA seems to transfer almost nearly naked fashion or through the protrusion-like structure. So, but now we speculate that this phenomenon might be mediated by dye. Then we set up similar experiments in which mouse ES cells express CD81, in this case colored yellow in this video, and mouse human ES cells have the magenta actin colored in magenta, and TFAP2C mRNA was tracked by the molecular beacons that bind mRNA specifically bind and mark with the fluorescence. And in this case, we colored the TFAP2C mRNA in light blue. And then here we can see the many vesicles are moving between mouse and human cells. And here you can see that mouse-derived vesicles, yellow vesicles, exist in the human cells, and these vesicles are accompanied with the TFAP2C mouse mRNA. And these vesicles are running on the actin filaments in the human cells and go and back between the mouse and human cells. So you can see the movement of these vesicles. Oh, yeah, these vesicles moving fast, and some vesicles moving between mouse and human cells, and some vesicles go to some kind of deeper side of the human cells. Here is a human nucleus, and some vesicles, some mouse vesicles reach deeper area of the human cells. So this result suggests that such direct contact dependent naive conversion is a kind of synchronization to naive status. So that phenomenon seems to be likely dye mediated. It is one of dye function, I suppose. So, in summary, here we demonstrated novel phenomenon to machineries, phenotypic synchronization of cell cycle and direct intercellular vesicles exchange dive and intercellular vesicles. So, of course, what we have shown here is mainly live imaging based, and much remains to be elucidated. However, as we discover more biological processes involving dive and we revealed molecular mechanisms of dive, significance of dive will be increased. So we consider that dive, that is a novel modality of cell-cell communication, could have a potential to a kind of that reshape our understanding of the biology. Okay, much of this work has done by Tomohiro Minakawa and with Mima Shikanai, and now continue his work. And we are also grateful to many collaborators for their contributions. Thank you for your attention. I'm happy to take questions.
Well, thank you very much, June, for that presentation and for sharing this very exciting work with us. I think that there are many, many questions, and I think people will probably have some right now, and there will be others that they'll be puzzling over the next few days as they remember your presentation. So let me start here. Let's see here. Let me just remind everybody, if you do have a question or a comment, please just put it in the chat box, and I will call on you. And I have now also enabled you to unmute and to share your video if you would like to. So let me start with Fabrice. Fabrice is here and has a couple of questions. I know that after you ask your question, June put up a schematic of how this could possibly be working, but there's probably more that you'd like to discuss in any case, and you have a second question too. So Fabrice, please go ahead.
Yes, thank you. That was a wonderful talk and beautiful images. Congratulations. Yeah, so actually that was timely. I was putting my questions when you proposed the hypothesis that there was some actin cytoskeleton being involved in the transport of the vesicles. Have you also considered tubulin and maybe like some of the motor proteins such as kinesin in the transport of those vesicles?
Yeah, thank you, Brett. So actually, in the tunneling nanotubes, so not only actin, the tubulin, and the kinesin system is used. So yeah, I of course consider that these molecules are also the candidates for the involved in the dive system. But mainly from that speed of that vesicle movement, it's very fast. And also we try to check that the stain that tubulin, but we've not obtained no apparent staining about tubulin. So now I consider the actin the main filament used in the dive, and also due to the speed of that moving, so myosin or some fast molecular motor would be used other than kinesin. So now we think that actin and myosin system is most likely system in dive.
Yeah, that makes sense. Yeah, that's very nice. Yeah, and so based on the images, I mean, obviously, you cannot record that many cells and time frame, but in terms of the numbers of those vesicles transported between donor cell and recipient cells. So do you think it's really like a tightly regulated mechanism and like really like a mechanism that is actively regulated, or more like a stochastic phenomenon, right? I mean, because based on the numbers that you seems to see during those transfer.
Yeah, it's a very, very good question, but actually, so we don't have an answer for that. But yeah, so we observed that after the co-culture of the cells, green and red cells, in some cells, so many vesicles move into some cells, but to other cells, almost no vesicles move into that. So there are some not completely stochastic, but some selective system or for the transfer. So would exist, but still we don't know.
Thank you. Perfect. I'm going to let the mic to other individuals. Thank you very much. That's very appreciated for your answer.
Thanks for the question, Fabrice. And we'll go next here to Anand. Anand, please let us know about your thoughts.
Yeah, thank you, Ken. Quick question. It's kind of follow up. Let me just turn off my mic. Okay, here goes. So yeah, sorry about these technical difficulties. I also had a similar thought about the cytoskeletal protrusions coming out from cells. So it looks clearly like they're making. I'm just kind of confused, like what kind of cell types are used over here? I mean, you spoke initially about mesenchymal cells, I guess, MCs, and then was
Okay.
stem cells.
We are used mainly in two types of cells. One is the mouse ear cells, and another is the MDCK cells. This is epithelial cells of the renal tubulus of the from dog cells, from dog.
Oh,
So these are very, I would say, well used epithelial cell lines. So, and most of the video we show later part of the presentation is the result of the MDCK cells.
okay. The reason why I asked is, is it possible to do a scratch assay on these two cells and show that like these cellular protrusions come from one cell to the other cell, so you can image them much better rather than like putting them as a like putting all of them together and aggregating them.
I've not done the such scratch assay, and we don't know about these cells from the protrusion well or not. But in our experiments, we culture cells very uh, dense culture condition, and these cells are attached directly. And I suppose in our cases, so almost no protrusion were formed.
Okay. A follow-up question is that so mo most of the images kind of showed that the intracellular vesicles shuttling from one cell to the other cell. So how hard are these vesicles? Do they fuse with the plasma membrane? They don't. I never saw them fusing. They were just like going back and forth. So do you think like they're really hard, like the lipid structure, and so they're not able to fuse to the plasma membrane, or is there any subtype of EVs that's actually doing this?
Yeah. So yeah, it's the same impression I have that many basically seems go in terms of go back and forth, and but I'll say some vesicles reach the deeper side in the human cells. For I show you that, and so yeah, two things is it requires. So I like to see that basically how vesicles pass through the plasma membrane. So we focus on like that area. So to to trace a longer time for that vesicle transfer, which required more wide view is required. So it's required so different condition to completely trace that vesicles, the result of the vesicle. So yeah, it's very important point and interest point, and yeah, currently we'd like to try to that matter, but now we could not get uh, uh, how solid result. So,
No worries. Thank you. For the great work, though. Thanks a lot.
Thanks, Anand. And next is Agnieszka, and please correct me if I'm pronouncing your name incorrectly.
No, that's perfect. Thank you so much for the beautiful presentation. So I have a very similar question to Anand. So I wonder, because obviously what you were showing may not be, it just looks like the vesicle is going through an opening in the membrane, but it may not be the case because it's just probably how we are able to see it. But I'm wondering, how do you envision if the vesicle is going inside, sort of invading the cell, what kind of machinery, trafficking machinery is it using? Because it cannot just be shot like a ball across the membrane and just randomly go into the cell. So it needs to take the trafficking machinery of the cell that it goes into. So how do you envision that this process is going?
So yeah, it's just a putative model. So now I think like that. So basically moving very fast and pass through the plasma membrane just in 10 seconds or so. So it's very rapidly pass through the plasma membrane. And uh, basically pass through almost the same position, same site again and again. So that means that there's special routes. So then we now speculate that systems like that should exist. There should some a pore-like structure that may be that would be generated by fusion on the plasma membrane. So and on the tip of the tunneling nanotube, there's cell fusion, and so they connected on the cytoplasm. So similar things occur in the plasma membrane. So then such pore-like structures are generated by the fusion of the plasma membrane, but still we don't have the exact evidence of that. And now we are trying to see that. And how say some fibers extend in them and basically moving using the molecular motor. So then the basically moving rapidly go through the plasma membrane and using the always going the same way, same route. So this is one of current our hypothesis.
Okay, so that and the openings in the plasma membrane are these. Are you assuming that the membrane is discontinuous and the vesicles are just finding way through sort of channels in the membrane, or there is a process, some signaling that in contact with the vesicle there is an opening, because that would require very complex machinery, or is it just a is it random or it is regulated?
Yeah, yeah, that's really important point. But yeah, just we start to observe these phenomena, and still we know anything about that. Yeah, of course, yeah, that's really important point. But in our observation, so basically touch the plasma membrane, but soon, just within 10 minutes, 10 seconds. So basically go through. So yeah, it's required more longer time to make the pore and after touching the vesicles. So I suppose that vesicles and the fiber rail is already exist and just basically go through that.
Okay. And just one, I don't want to take too much time, but just one last thing that I want to make sure that I understand. So you're proposing that the myosin fibers or actin fibers are being exchanged between two different cells.
Yeah, also that's important point. But yeah, how actin fiber generated between cells and how the I think that motor molecule, I think, can be traversed from to the another cells. That would be possible. And how actin fiber generated between two cells is currently I don't know. So two fiber join the cell membrane position one, some one actin extend to another cell. Still we don't know. I'd like to.
And you don't think that there is a possibility that there's actually a process that maybe it's not being observed because it's so fast that instead of just membrane, actually vesicle being shot, like it's shooting into the cell, that there is an exchange of the content and fusion of the membrane of the vesicle at the plasma membrane, and then it is its own cell that is packaging the vesicle again, the content as a vesicle and going in. So do you have any data that will support or disqualify this argument?
But yeah, vesicles is always identically exist. So we never observed the fusion to the plasma membrane.
Okay. No, thank you very much.
Thank you, Agnieszka. And also I see that Fabrice is saying maybe a flip-flop mechanism of the PM that enables fast vesicle internalization. But yeah, that's I guess that's what you've just addressed, June, and that you I think what you're saying is you don't see the mixing of the the colors, correct?
Yeah. So, but for example, so there is a vesicles with, for example, green vesicle covered by red vesicle. Such vesicle exists. So this such vesicle would be transferred not through dye, but endocytosis or a conventional mechanisms. And our experiment is just co-culture. So some EVs go out and enter into the adjacent cells through the endocytosis. So in adjacent cells, we actually observed that double colored vesicles. But now, in our video, so these transferred vesicles are just independently green, never accompanied by the red plasma membrane components. So I think that through the dive transfer, dive phenomena, so vesicles independently pass through the plasma membrane, not mediated by the endocytosis.
Thank you. So, Satoru, you introduced these concepts of naive and primed cell. Could you tell us? Could you explain what you mean by that? I like this idea.
Thank you, Chris.
Thank you for such a beautiful work and also a lot of entertaining videos. I'm kind of new to this field, so that maybe my question could be not make sense. But I wonder, I had some impression that entire story kind of give this EV transpassing is kind of bidirectional. It's moving forward, back and forth. But the last data showing the EV actually the cell synchronizing from co-culturing naive cell, naive is S, and then primed ES, but the synchronizing to the naive phenotype. That's wondering why, because if they so that's why I think this is kind of one directional EV transpassing, or is this some mechanisms?
Yeah, I suppose bidirectional transfer may occur, but in our experiments, we only label the vesicles only in the mouse cells. Human cell uses the standard actin filament. So in such experiments, we didn't see that vesicles from human cells. So we cannot detect that vesicle transfer from human to mouse in such experiments. So yeah, if we use that human cells that map the vesicles, yeah, I think it would be possible that we will see that the vesicle transfer from human cell to mouse cells.
Okay. So, but if that's the case, that's transfer from the human to the mouse cell does not impact on the phenotype.
Yeah, I don't know. That is from the other group, so we don't know about that. But now, yeah, but actually, for example, so we performed that synchronization, the differentiation at that time. So in that experiment, we used the forced expression of PK. That's very strong condition. So then how say slowly differentiating cells synchronizing the past differentiating cells. But if the house more physiological conditions, so such synchronization occurs both ways. So bidirectional exchange may occur, and so synchronize into not only to the one direction, both synchronize to one of the intermediate state. So I think that such bidirectional synchronization would occur, I suppose.
Thank you. I see. So in general, the physiological condition, they are more really exchanging the information about EV. But this is very experimental, so you can force it to emphasize one direction. I think. Thank you.
So this talk of directionality and also the observations of the fusion, the lack of fusion of some of these particles that are transferring makes me wonder too about other tasks that these particles could be performing, and specifically things that would involve the surface of the extracellular vesicle, or the vesicle, I should say. I don't know whether we should call these EVs, but are they grabbing something from a recipient cell and bringing it back into the source cell without necessarily loading into the lumen. Lots of questions that were raised for me through this presentation, and I'm sure for others as well. But I think that brings us to the end of the questions that are in the chat box. No, I spoke too soon. Beta, you have a last minute question here. Please go ahead.
I'm sorry to be.
No. This is all about questions.
I want to ask about the cargo delivery. If the EV is fusing with the plasma membrane, then the cargo will be delivered straight into the cytoplasm. If the EV comes as whole into the cytoplasm, then do you think that there is a special organelle or a special target where actually the EV will continue and release on the proper spot the cargo, or because it can go, it can go into the endosome and then just be destroyed. And
it's also very important point. So but currently, so I try to identify the true structure of the inter-CVs, and we perform the PIPSEM, and inter-CV-like structures we identified, but we not know that these are actually moving vesicles or not. But these candidate vesicles single membrane, so just like the endosomes, like that, not a double membrane. So just single membrane they have. So I don't have any evidences, but I suppose that these vesicles behave like the endosome and Golgi vesicles or such. So single membrane vesicles is resemble to our intercellular CVs, intercellular vesicles. Currently, I don't know how these vesicles behave that are processed after transfer to the adjacent cells. It's issues I have to elucidate after that. Thank you.
Thank you.
So we have plenty of subjects here for further investigation. So thank you, Jun, for sharing this with us and giving us plenty of food for thought. And I'd like to thank everybody who has joined this session too, and contributed your comments and your questions. I'd like to invite everyone to please also join us next week. We're going to have a session on membrane dyes, or I should say lipid dyes, and measurement thereof using some single particle techniques. And if you have any suggestions for future EV clubs, please do reach out to me, and we can see if we can get something scheduled. So thanks again to everybody. Hope you have a good rest of your day, rest of your night, wherever you are. And I hope to see you on an EV club again very soon. Bye now.
Thank you. Thank you. Bye.